Okay, let's have a look at gene regulation here. Now this is an area that we know quite a lot about in bacteria, but it is an area that's quite complicated, and I suppose it's an area that scientists are still learning quite a lot about. I suppose the basic principle is that in any particular cell, we don't need all the genes all the time. So there's certain genes that are activated, but more particularly, we can switch on some genes when we need them and switch off some genes when we don't need them. Some genes you might need to produce proteins continuously, but there's probably a vast majority you only need to produce the proteins or enzymes at particular times. And that's what we call gene regulation. If we look at a molecule here, we can imagine that our DNA molecule that winds around here is coiled up or supercoiled to form our chromosomes, and there'd be particular areas along the DNA that code for particular genes. And what we're talking about here is that maybe one area here might have a gene that might help regulate a gene in another area. And that's what we'll look at. So while we look at this, it's probably important to remember that this is related to the processes of transcription and translation, because it's these genes that are producing a particular protein, and quite often a functional protein, for example an enzyme. If an organism was to produce a particular characteristic, it might need several genes in a row to produce the enzymes to make that particular metabolic pathway happen. So quite often an organism would read these genes continuously, one after another. Now in an organism we quite often have a regulatory sequence of genes, and this sequence is called an operon, and it has a few particular genes involved. It has a promoter gene which starts a regulation with RNA polymerase. It has an operator gene, which is basically the on-off switch where the gene can be controlled, and it has structural genes because quite often there's a few genes involved. There's also a regulatory gene which sits outside of this particular operon or sequence. It sits on another part of the DNA, and that can control repressors. So let's have a look at this in some more detail. So this diagram is probably a little bit clearer about what's going on. This part here is the promoter, and the promoter is where this molecule here, the RNA polymerase, attaches, and it runs along the DNA strand making RNA, making messenger RNA. Okay, so the RNA polymerase attaches at the promoter, the particular sequence that allows it to attach. Just following this is an operator gene, and that's basically the on-off switch, and that can be switched on and off by repressor proteins. And following this is our structural genes, and we quite often have a number of genes involved: gene A, gene B, gene C, because to make a particular characteristic happen, you quite often need a few genes to make a number of different proteins or a number of different enzymes. The last part we have is a regulatory gene, and this can be either upstream or downstream of the DNA in a different part of the DNA, and that produces a particular protein, and this protein is a repressor protein, and that fits in when that repressive protein sits in the operator gene, it switches it off. Okay, now it's important to understand that control happens outside of this operon, and that's what we're calling gene regulation. It's also worth considering that this repressor protein can be under both positive and negative control. It can be under positive control if a particular chemical is present. It could fit into the repressor protein and change the shape of that repressor protein, meaning that it doesn't fit into the operator gene, which would allow the RNA polymerase to read to travel along and read the particular genes. It could also be negative control, where when a particular chemical is present, it fits with the repressor protein, and it then does fit into the operator genes, switching it off, stopping RNA polymerase traveling along and reading the genes. So let's have a quick look at an example of this and have a look at how regulation could occur in bacteria. And this example we're going to look at is regulation that's controlled by lactose. So in our example here, we have a regulatory gene which is somewhere else producing a repressor protein. We have a regulatory sequence which is a promoter, an operator, and a whole lot of genes that have to do with lactulose. And normally, these regulatory genes are producing this repressor protein, which is sitting in this operator gene, sitting in the on-off switch, switching it off, stopping the rest of these genes being read. These genes don't need to be read most of the time because we don't need to produce enzymes to break down lactulose if there's no lactulose present. Now what happens is when lactose is present, it fits into this repressor protein, changing the shape of it so it doesn't fit in the operator gene and doesn't block the production of the lactulose genes. So what can happen now is that RNA polymerase can run down the DNA strand, producing the enzymes that will break down lactose. The cool thing about this is when all the lactose is broken down, there's no longer lactose molecules to block the repressor protein. So when all the lactose has been broken down, the repressor protein can then fit back in to the operator gene, blocking the production of the enzymes that you don't need. Okay, a really effective way for a cell to switch on what it needs on and off. Now, although this is a complicated example, things are quite a lot more complicated in eukaryotic cells. We've just been looking at a prokaryotic cell. We've been looking at the lactose regulation in a prokaryotic cell. In eukaryotic cells, we know that it's involved. There's lots of genes that are involved in regulating transcription. Quite often, the DNA folds back in on itself to switch things on and off and to control those sequences. And it's still something we're learning quite a lot about. So good luck with learning about gene regulation.